The value of synapses that can handle chemical signals becomes clear when we consider how they function in the context of the nervous system. A huge network of neurons, interconnected by many branching pathways, performing complex computations, storing memories, and generating plans for action. To carry out these functions, neurons have to do more than merely generate and relay signals. They must also combine them, interpret them, and record them. Chemical synapses make these activities possible. A motor neuron in the spinal cord, for example, receives inputs from hundreds or thousands of other neurons that make synapses on it. Figure 1245. Some of these signals tend to stimulate the neuron, while others tend to inhibit it. The motor neuron has to combine all of the information it receives and react either by firing action potentials along its axon to stimulate a muscle or by remaining quiet. This task of computing an appropriate output from a cacophonous babble of inputs is achieved by a complicated interplay between different types of ion channels in the neuron's plasma membrane. Each of the hundreds of types of neurons in your brain has its own characteristic set of receptors and ion channels that enables a cell to respond in a particular way to a certain set of inputs and thus to perform its specialized task. Furthermore, the ion channels and other components at a synapse can also undergo lasting modifications according to the usage they have experienced, thereby preserving traces of past events. In this way, memories are stored. Ion channels, therefore, are at the heart of the machinery that enables you to act, think, feel, speak, and perhaps most important of all, to remember everything you read in books like this one. Essential concepts. The lipid bilayer of cell membranes is permeable to small nonpolar molecules such as oxygen and carbon dioxide, and to very small polar molecules such as water. It is highly impermeable to most large water-soluble molecules and all ions. Transfer of nutrients, metabolites, and ions across the plasma membrane and internal cell membranes is carried out by membrane transport proteins. Cell membranes contain a variety of transport proteins, each of which is responsible for transferring a particular type of solute across the membrane. There are two classes of membrane transport proteins: transporters and channels. The electrochemical gradient represents the net driving force on an ion due to its concentration gradient and the electric field. In passive transport, an uncharged solute moves spontaneously down its concentration gradient. A charged solute, an ion, moves spontaneously down its electrochemical gradient, and water moves down its osmotic gradient. In active transport, an uncharged solute or an ion is transported against its concentration or electrochemical gradient in a process that requires energy. Transporters bind specific solutes, inorganic ions, small organic molecules, or both, and transfer them across the lipid bilayer by undergoing conformational changes that expose the solute binding site first on one side of the membrane and then on the other. Transporters can act as pumps to move a solute uphill against its electrochemical gradient, using energy provided by ATP hydrolysis, by a downhill flow of Na+ or H+ ions, or by light. The Na+K+ pump in the plasma membrane of animal cells is an ATPase that actively transports Na+ out of the cell and K+ in, maintaining the steep Na+ gradient across the plasma membrane that is used to drive other active transport processes and to convey electrical signals. Channels form aqueous pores across the lipid bilayer through which solutes can diffuse, whereas solute transfer carried out by transporters can be active or passive. Transport by channels is always passive. Most channels are selective ion channels, which allow inorganic ions of appropriate size and charge to cross the membrane down their electrochemical gradients. Transport through ion channels is at least 1,000 times faster than movement through any known transporter. Other channels conduct water or other small metabolites. Most ion channels are gated. They open transiently in response to a specific stimulus, such as a change in membrane potential, voltage-gated channels, or the binding of a ligand, ligand-gated channels. Even when opened by their specific stimulus, ion channels do not remain continuously open. They flicker randomly between open and closed conformations. An activating stimulus increases the proportion of time that the channel spends in the open state. The membrane potential is determined by the unequal distribution of electric charge on the two sides of the plasma membrane and is altered when ions flow through open channels. In most animal cells, K+ selective leak channels hold the resting membrane potential at a negative value, close to the value at which the driving force for movement of K+ across the membrane is almost zero. Neurons propagate signals in the form of action potentials, which can travel long distances along an axon without weakening. Action potentials are usually mediated by voltage-gated Na+ channels that open in response to depolarization of the plasma membrane. Voltage-gated Ca2+ channels in nerve terminals couple electrical signals to transmitter release at synapses. Transmitter-gated ion channels convert these chemical signals back into electrical signals in the postsynaptic target cell. Excitatory neurotransmitters open transmitter-gated channels that are permeable to Na+, and thereby depolarize the postsynaptic cell membrane toward the threshold potential for firing and action potential. Inhibitory neurotransmitters open transmitter-gated Cl- channels and thereby suppress firing by keeping the postsynaptic cell membrane polarized.